and I'm going to introduce the next two tutorials, tutorials 3 and 4, on networks of neurons with different types of cell models. So tutorial 3 is about multiple biophysical neurons and networks constructed from them, and tutorial 4 sort of follows quite similarly to the material in tutorial 3, but in place of biophysical neurons, it uses GLIF point neuron models. And so just a couple general points that apply to both of the tutorials. A lot of the material that is coming in these early tutorials, the ones that I'm going to be giving and the next two coming up, these are sort of building incrementally towards some of the larger network models that our group has developed, such as the full V1 model in the Billy paper. So if you're someone who kind of likes to see the the big picture first, and then the path to get to it, you might want to go through this paper and sort of get a bit more context for some of the things that come up in these tutorials. And then another point is that even though these tutorials, most of them have the cell models kind of pre-populated, the components folders pre-populated, and a set of neurons selected from the cell types database, I'd really encourage you to actually go to the cell types database and try downloading another model or replace existing models in the tutorial material, because I think making sure you know where different pieces go, any issues that might come up when you're trying to replace one for another or add more cells. I think these sorts of things will really help you with learning this material if you haven't been using these before. So let's go through tutorial 3. So the sort of overall goal of this model is that we're creating a network with 80 cells. And these 80 cells will consist of excitatory and inhibitory cells, biophysical cells, the ones you just heard about in the last tutorial. And these will be constructed based on 3 cell templates. So 3 of the cell models from the cell types database for the excitatory cells will be used to create a population of 60 excitatory cells, and a single inhibitory model of a PV cell will be used to create the inhibitory cells of this model. And sort of the general flow of both tutorial 3 and the next one is that we're going to be constructing the nodes. There's going to be some instructions and visualizations created from VND to look at what's been created, the nodes, and then the networks after the edges after they've been constructed. And then the model will be simulated. We'll see the output. And we can do some comparison between the different model types. I'm not going to say too much about the VND visualization that's kind of peppered throughout these tutorials. I kind of leave it up to you whether you want to go through that now as you're working on these tutorials in the afternoon, or whether you want to kind of save those parts of the tutorials for after Barry gives his talk on VND tomorrow. You could come back to the models that you've constructed and then start kind of exploring them in 3D. So that's at your discretion. Okay, so I'm going to just run through the node creation for the excitatory cells created from one of the 3 populations of excitatory cell types in this network. And lots of the details here you've already heard, but it probably wouldn't hurt to to hear some of these details repeated. And if something is unclear, please jump in. And also, because all this material will be shared with you, and the tutorials themselves have a fair bit of description of what's going on, don't feel like you need to be jotting down notes about everything. I think it's probably more helpful just to get an idea of what is going to be covered in the tutorials, and then just kind of go through them kind of fresh after lunch. Okay, so the first step is we're instantiating an L4 network, layer 4, using Network Builder. We're calling the add nodes method of our L4 network to create our first set of cells, and these are going to be the 20 excitatory cells constructed from this first cell model downloaded from the cell types database. So our n is equal to 20 here. There's a number of directives to BMTK that you heard about in the last tutorial. The model type, these are biophysical as before. Model template, the cell types database, biophysical type 1, Hawk file, model processing, dynamics params, and morphology. You've already heard about this. Next, we're going to be applying a rotation to each of these 20 cells. Oh, sorry, before that, we're defining the coordinates. And so the convention that's being used in a lot of the cell models here and the different data sets at the Institute is that the XZ coordinate system is parallel to the peeled surface, and the y-axis is the depth spanning the cortical layers. So just something to keep in mind. And now, so this this cell model that we're using, it's a pyramidal cell, so it has an orientation. It has apical dendrites that project up to superficial layers. And something that's necessary for all these models is, my cursor showing up, we're applying a rotation to the z-axis to kind of straighten each of those cell models up, so it's transverse to the peel surface. And then
how do you know that's necessary?
It's just, if you look at the models, you'll see that the way that they were, the way that the morphologies were captured from the cell types database, has.
Visualize it, you can see it basically.
Yeah, and I think it's probably going to be a little bit different from one cell model to another. Yeah, another good reason to to look at what you're building as you are constructing your models. But so in addition to that sort of correction, we're also applying a random rotation about the y-axis to get some heterogeneity in terms of the cell morphologies. So recall that these 20 cells we're creating all have identical morphology, but sort of the least we can do is kind of give them a little different rotation to create some variety. So a few other things to note here. So unlike before, when we just had a single cell, we're now dealing with a set of 20 cells. And so there are two main ways of specifying parameters or properties of nodes in the model. So the properties can either be set at the cell types level or the node types level, or the node or cell level. So if you give a single string or tuple or a single value, that value is going to be applied to all 20 cells identically in the nodes that you're creating. Whereas if you apply a list, which is what's being done when we're setting the locations, or for the rotations, if a list is provided and it needs to have length equal to the n of the nodes being created, these are set at the node level, not node type level. And so this is sort of a key thing to keep in mind as you're constructing networks of neurons, with an n greater than 1. And then, so as before, there are also optional parameters. So these don't directly do anything in terms of the construction of the nodes themselves, but these are useful properties to have that we will utilize in constructing the edges. So for our excitatory cells, we're assigning a tuning angle property. So the idea here is that this network that we're creating is sort of meant to be a sort of a toy model of the visual system, and these cells have preferential responses to moving gratings, oriented in different directions, for example. And so to wire up our network in such a way that these sorts of properties might emerge, we assign these properties that we then use in our wiring up of connections. So, okay, so we do this for this particular cell model, and then again, identically to this for the other two excitatory cells that we've downloaded from the cell types database, and again for the inhibitory cell. But the only difference for the inhibitory cell is that, well, first the EI type is, we're denoting it as I for inhibitory, and we're also not assigning it a tuning angle. And this is just meant to represent or to reflect the lack of connection specificity for PV connections compared to excitatory cell connections. Okay, so next we're creating edges, and for the recurrent connections in this network we've created, so suppose that we've created our 75 cell network, 80% excitatory, 25% inhibitory. We have to start wiring it up. And the way that we do that is first by utilizing for the source and target population, we're going to do this first for the excitatory to excitatory, then excitatory to inhibitory, inhibitory to excitatory, etc. So I'm just going to go through one example, excitatory to excitatory. We specify our source and target, and we define a connection rule that is going to take all the properties that we've defined for the pre and post synaptic populations and return the number of connections, 0 or more, between that pre and post synaptic putative connection. We're also applying or defining a synaptic weight that's going to be shared by all of the edges in our network, and likewise for delay and the dynamics parameter and the model templates. So these are shared by all of the E-to-E connections that are being defined by this add edges call. Any questions up until now? So yeah, below is our custom excitatory to excitatory connection rule. So they should return a single integer, 0 if there are no connections formed, 1 or more if there are one or multiple connections. So in this case, we're extracting the tuning property that we defined randomly for the source and target cells, and we're defining, we're determining the circular distance between those properties, and we're returning connections based on that similarity or distance in their respective tunings. Now, for assigning, so when we were creating nodes with for an n greater than 1, I mentioned that we can either assign a string or single number or a tuple that's shared by all of the nodes, or we can assign a list of length equal n that allows us to specify a distinct value for each of the nodes. Now for edges, it's a little bit more tricky because we don't initially know how many edges there are going to be. For example, in our distance-dependent connectivity rule, we don't know a priori how many connections there are going to be for the EDE cells. And so what we do here is we're using this add properties method, which allows us to define a set of properties and allows us to specify a rule or use one of the built-in rules that consider that assign those properties for each of the edges that have been constructed in our network. And so what's being done here is we're defining details of where those connections should be assigned on the morphology of these cells. And this is based on the SWC based definition or characterization of these cells' morphology. And we're assigning a random synapse location on either the basal or apical dendrites within some range of distance. And this is part of a built-in RANSYN locations function. Any questions at this point?
I had a question about the previous slide. So when you specify the distance range, if that exceeds the extent of, say, the dendrite, what would you do if the random number generated a distance where there's no number?
As far as I know, it just selects actually Kale, does it
Range?
yeah, if the range is outside of the let's say the most distant portion of the apical dendrite, is it going to,
If the minimum range, if the min,
let's say the max, let's say here's our dendritic tree and we're going up to this range, is it going to assign?
it's essentially 0 to infinity, it will get every single possible dendrite. It's not going to choose something at like near infinity.
Are you saying it does it rescale to the available length, or does it? What happens if like the assigned location is outside of the actual morphology? Is a connection still formed?
It won't.
It won't. It'll return
If
a.
you go from like 0 to like say 2 million, so you notice sometimes we'll put like 1 to the 10 to the 20th, which is a hugely large number. That just means choose any the option is to choose between any possible available axon or segment, or not axon, dendritic segment.
Okay.
If you go between like a million microns to 2 million microns, you're going to get no segments at all.
Okay. So if it chooses a point that's outside, like within some finite range, it'll just return 0 connections
It's
there.
gonna find all segments within the range and randomly choose those. That's what.
I see.
I see.
Okay. So there have to be some portion of the cell morphology within the range that's specified. Any other questions up until this point? Okay, so now we have our network, excitatory and inhibitory cells. We've connected them together. I've skipped over the other types of connections, but it basically follows what we did for the excitatory to excitatory cells, except other connection types aren't going to have the same orientation dependence. But our network is wired up. But now we need some drive to our layer 4 V1 toy model. And we're going to create something that, very abstractly represents thalamic input coming from LGN projecting to our network. And so what we do is we assign an XY coordinate to each of the LGN cells in our feed-forward input layer. And both these positions, these XY positions, as well as the XY positions in our V1 cell model have shared what's the right word? Mapping of the visual space, into the physical space of coordinates that we've assigned them. And what we're going to do is, for the projections of each of the LGN cells, we're going to look for an ellipse of some specified size. We're going to look for all the cells within that region and assign connectivity to those eligible connections based on another custom-defined connection rule. And in subsequent tutorials, this input is going to be kind of built up to something significantly more closely resembling the actual LGN. But for now, we're going to just have random spiking from this LGN layer driving our layer 4 model. These LGN cells are basically just placeholders to relay spiking inputs to our layer 4 model. So these are called virtual node types, so they don't have any intrinsic dynamics. If you're familiar with Nest, these are like parrot neurons. They just relay spiking input to downstream targets. We connect these up as I described. We're going to create spikes for LGN, and this is going to be the drive to our network. And then we're going to simulate the system. Let's just quickly take a look through the code defining these connections. So first of all, we've created an LGN network. We're adding edges to the LGN network, and that is going to have a source and target. Something new that's happening here compared to the connections that we formed between the recurrent connections is rather than iterating through all individual pairs of, for example, EDE cells, as we did for our EDE connections, we're using a different iterator here. We're using a one-to-all iterator specified here. The default is one-to-one. We didn't specify that earlier, but it was using the one-to-one iterator. So what's happening here is that each of the LGN cells is going to be considered in turn, and our connection rule that we define is going to return a list with length equal to the number of cortical cells in our model, 80. And for each of those, it's going to return an integer, 0 or greater, specifying the number of connections between that LGN cell and each of the targets. And because of the rule that we're defining, we're only going to have non-zero values when those cortical neurons are within this this ellipse that we're defining here. So, yeah, try to read through the logic of this connection rule, see if it makes sense as you're working through the simulation, and definitely, once you're using VND for visualization, come back and take a look at some of the connections formed from a single LGN cell to targets and just verify that it kind of matches your reading of the code. Okay, there's an optional portion of this tutorial, and again, this also relates to a component of the full V1 model described in the Billy paper that I mentioned. And so the issue is that, our initial network sits within this cylinder, and we have distance dependence underlying connectivity of the cells, and so there's going to be edge effects. The cells on the periphery of our column or our cylinder are going to have fewer connections than the ones in the center. How can we mitigate this without creating something that's overly computationally intensive? Well, one way is to create a population of simplified integrate and fire cells in an annulus around the periphery of our cylinder, and these will be connected to and from the actual biophysical cells in our model. And this will not completely resolve the effect, but it should lessen it. And so towards the end of this tutorial, you can kind of work through the steps of that if you're interested in this issue. I won't go through the adding of nodes and edges again. We've been through that a few times. Once you have created the Sonata files for this additional network component involving these integrate and fire cells, you can either rebuild the full config file, or you can just add the necessary new components. And again, this is sort of one of these things that's helpful to, to kind of add in manually just to get a sense for how these things are constructed. So here we're adding in a new set of nodes for the lift network, and we're adding in the relevant edges. Okay, a few other additional tutorials and material related to this. So there's also a section in this tutorial that, again, dives into the Sonata file format and exactly how nodes and edges are stored as HDF5 files. So if you really want to get into the nuts and bolts of things, you should work through this. And there's another tutorial in a separate notebook, tutorial 3A. Beatrice mentioned this. And this goes through the SWC file format. And it's useful if you want to get a better sense of how the connections are being assigned in these morphological cells with these custom connection rules. You really should have a sense of how these are constructed, and it's pretty straightforward. Okay, this next section is very short because one of the beauties of the Sonata file format is that our networks are interchangeable. We can kind of reuse large blocks of the network file data and replace only limited portions that are necessary, and move this network from something that runs with a neuron backend to something that runs with a nest backend using completely different cell models. So again, the first step is that we're identifying or choosing the cell models we want. In this case, we're using these one of these GLIF types that are also available from the Allen Cell Types database. At the end of this tutorial, I have a couple of references. One of them is for the nest doc page for these different model types. So again, we've chosen a couple for you, but please try replacing things. See what how differently the network behaves once you have it running. Okay, and now to construct our GLIF version of the same network type, we have two choices. So we can basically build everything again from scratch, or we can reuse existing Sonata files that we already created. If you compare the construction of the biophysical to GLIF, they're quite similar. For the GLIF, it's not necessary to assign the rotations, because these are just point neurons. And it's also not necessary in the edge definition to assign the actual location of the connections, because again, these are point neurons. So in terms of the node type files and the edge type files, again, there's a lot that's reused. Here, the model type, has been changed from biophysical to point neuron. There's a different model template and dynamics parameter, but these are shared between lots of the cells or lots of the nodes in the network. And then for the biophysical cell type, there's a number of additional properties that aren't required for our GLIF network that we can just remove or ignore. And same idea for the edge types. One important point here is that the synaptic weight, because these are different cell models, there's a different meaning in what unitary synaptic weight is between the point neurons and the biophysical cell models. That depends on sort of the history of how these model formats were constructed. These values don't correspond to one another. So just something to keep in mind as you're trying to convert, one model to a corresponding second model of a different type.
What's the difference between a static synapse and the EXP? I know EXP just seems the double exponential. How is that different from the static synapse?
So the static synapse type for the GLIF neurons, it just means that there's no SDP. It doesn't change over time. I don't think there's any other details carried with that. The dynamics params itself defines the parameters related to the synapse, and the synapse itself is defined as part of the model template itself. So the actual cell model itself in NEST has a particular synapse functional form associated with it. In this case, it's so up until the end, it's the cell model specification, and then postsynaptic current means it's a current-based synapse.
I have a question about the weights. So when you say that there's some tuning of the weights that's necessary if you go from the biophysical to point neuron, do they stay in proportion roughly, or do we have to tune each of the weights?
So with the biophysical cell model, these are going to be located, at various different locations. There may or may not be active currents, on that morphology. So, it's really going to depend completely on the details of the biophysical cell model. So I think I've covered most of this. There's just a few details on the iterator type. So we've used two of these. The first one we didn't explicitly mention. Again, here's sort of some initial output for these two model types. I could leave it as an exercise to you to see if you can adjust the synaptic weights in the GLIF model to try to bring things more closely in alignment. Again, just another reminder, there's a lot of good content here in terms of visualizing the network and connections that we've made. So if you don't kind of dig into this with VND on your first pass, definitely come back to it after the lecture tomorrow. And then just sort of to give a taste of things, here's a movie of the network we've created, rendered in VND for the point neuron model, showing activity over a 3-second simulation. Okay, yeah, so that's basically it. What are the next steps? I think the main thing is that it'll be interesting to see, how this toy network behaves with more realistic input, and that'll be the focus of the first tutorial tomorrow on FilterNet. And then finally, here's first is a paper that describes the different GLIF neuron models that are used both for the V1 model and throughout the Allen cell types database, and also a link to the NEST documentation for these Allen Institute GLIF models. So, thank you.